Hello again, class. It's the week of September 12th, and this week we're covering chapters 5 through 6. As usual, we will just be covering selections. Chapter 5 talks all about DNA, gene expression, and biotechnology. We'll focus mostly on DNA and biotechnology. And the big question is, what is the genetic code and how is it harnessed? Up at the top are the sections that you should be responsible for reading: 5.2 through 5.7, 5.9, 5.11, 5.13, 5.15 through 5.17. You need to know what DNA is and what it does, the process of gene expression involved in transcription and translation. I want you to know what transcription and translation are in general and where they take place in the cell and their purpose, but you don't need to know the specifics of the process. Explain the causes and effects of damage to the genetic code, so mutations, what are they and what do they do? We will talk about biotechnology and agriculture, and we will very briefly talk about its implications for human health. So what is it? What does it actually do? The importance of DNA goes far beyond its function as an individual identifier or by making our own cells, our own bodies, our individual self. Complex human characteristics and traits are linked to genes, and many have yet to be discovered. So you are responsible for this chapter. It just kind of told you that DNA is a molecule that all living organisms carry in all the cells of the body, and that it contains instructions for the function of every cell. So I would like to ask you, do all cells contain the exact same DNA? And the answer is yes, they do. And how do cells know to look and act different? Okay, so I have a brain cell versus a liver cell. If they have the exact same DNA, why is it that they look different and have such different jobs? We will discuss that in the chapter. The DNA molecule contains instructions for the development and functioning of all living organisms, so all life on Earth depends on DNA. And beginning in the 1900s and continuing through the early 1950s is when we began to learn about DNA's importance. So it wasn't until then, which is pretty crazy. So anyway, between 1900 and 1950, a series of experiments revealed two important features of DNA. First was that molecules of DNA are passed down from parent to offspring. So you got all of your DNA from your mom and dad. Second, the instructions on how to create a body and control its growth, development, and behavior are all encoded in the DNA molecule. These are two famous people, Watson and Crick. They, along with Frances Rosalind, who is often looked over, but she was just as influential as these two gentlemen in figuring out the structure of DNA. And its structure has a lot to do with its function. That's why we need to know something about its structure. In addition to Watson, Crick, and Rosalind Franklin, there were many formidable scientists that tried to determine the chemical structure of DNA and understand how it was assembled and shaped so that it could transmit so much information. So American Linus Pauling won a Nobel Prize in chemistry for his work on elucidating the structure of molecules when he began investigating the structure of DNA. But at the same time, Watson and Crick and Rosalind Franklin in England devoted their research to the tasks as well, and so they used X-ray pictures. Rosalind Franklin did the X-ray pictures of DNA that Watson and Crick then used to come up with their DNA model that's so famous, and it's shown here in this picture. But as soon as they figured out DNA structure, the answers to several other problems in biology, such as how DNA material might be able to replicate itself, became apparent. And we'll explore that process of DNA replication later in the chapter, but first we need to examine the structure of DNA in more detail. Alright, what is DNA? It's a macromolecule, which just means it's a big molecule. It consists of individual units called nucleotides. You need to know that. Each part of a DNA macromolecule is made up of nucleotides. Just like individual Lego blocks make up an entire Lego building, Individual nucleotides come together to make up the entire DNA structure. And then within the nucleotide, we have even smaller parts. So there's 3 components in each nucleotide, so that's shown up in the upper left-hand box. You have a sugar, which is deoxyribose, which you don't necessarily need to know, but that's what the D in DNA means, is deoxyribose. And then you have a phosphate group, which is important, and a nitrogen-containing molecule called a base. So the example in the top left box is adenine. So I want you to know the 3 components are a sugar, phosphate, and a base, but the bases are the most important. We'll cover those the most. And that's because the base pairing is what causes that double helix structure. So if you look now down, it says base pairs, that box. These DNA bases, there's 4 different types of them. I want you to know the 4 types. They're adenine, represented by a big A, thymine, represented by a big T, guanine, represented by a big G, and cytosine, represented by a big C. Those 4 bases bond to each other, and you need to know that A always bonds to T, and G always bonds to C. Okay, you won't get any other combination. A goes with T, G goes with C. And they bond together through hydrogen bonds. So know that these are hydrogen bonds. So these hydrogen bonds between base pairs A and T and G and C respectively form the rungs of our DNA ladder. You can think of it as rungs on a ladder. The backbone of the ladder is those sugar phosphates. So that's the backbone, but that's pretty much all you need to know about that. But remember those hydrogen bonds. Can't get away from bonds. Each sugar always has a base attached to it, and like I said, there's 4 different bases. And then on the other side of the ladder is going to be your sugar phosphate groups with another base, and so that's where they come together in the middle, forming those A's and T's and G's and C's. So you will always have the same number of A's as T's in a piece of DNA, and you would always have the same number of C's as G's in a piece of DNA. And remember, those are bases of your nucleotides. Alright, so ask yourself this question, see if you can answer it. Base pairing using what type of bond holds the 2 strands of the DNA double helix together? Is it covalent, ionic, hydrogen, or none of these options? The answer, of course, is hydrogen. Okay, which of the choices below will base pair with the following sequence? I suggest you get out a piece of paper if you need to and write these out, and then pause the video right now before I tell you the answer. But the answer is number 3, right? So that GAATTC. So that G will base pair with the C in number 3, and then that AA will base pair with the TT in number 3, and then that TT base pairs with the AA in number 3, and then the C base pairs with the G in number 3. Because G always goes with C, and A always goes with T. Alright, DNA is a nucleic acid, which is a macromolecule that stores information. Important information. It consists of individual units called nucleotides. And then those nucleotides are made of sugars, phosphates, and the nitrogen-containing base. So when we talked about the biomolecules and how there's 4 of them, we talked about lipids, carbohydrates, proteins, and then the 4th one is going to be the nucleic acids. We didn't cover those. We skipped those sections, and that's because we were going to be talking about that biomolecule here in chapter 5. So that's the 4th one. The DNA structure is like a twisted ladder. It's a double helix, and the sugar phosphate groups are the backbone with those base pairs forming the rungs. And the sequence of bases on one side of the ladder is always going to complement the bases on the other side. So you have complementary base pairing. That is so important for how DNA works. Now that we talked about DNA as a whole, we want to talk about genes. So genes are chunks of DNA, and they're important because each gene contains instructions for how to make a protein. And proteins are what do all of the actual stuff in our bodies. Okay, so the genes are holding the recipe, but the proteins are doing all of the work. DNA embodies the instructions for building the cells and structures for virtually every single living organism on Earth. All organisms use this same molecule to transmit this information to their offspring. So you can think of DNA like a universal language, and the letters of the language are A, T, C, and G. You can think of an organism's DNA like a cookbook. Just as a cookbook contains detailed instructions on how to make different foods, an organism's DNA carries the detailed instructions on how to build an organism and keep it running. Okay, so your whole cookbook would be all of our DNA, and then an individual recipe would be like a gene of our DNA. Just as a book can be viewed as a sequence of letters, with the book's meaning determined by which letters are strung together, a molecule of DNA can be viewed as a sequence of bases. So those bases would be analogous to individual letters in that cookbook, forming the words that we can read to make our different recipes. Because in a book, letters don't have much meaning on their own. When they are put together into words and sentences, however, their order holds a great deal of information. Similarly, the sequence in which bases appear in a molecule of an organism's DNA makes up a secret code that holds the detailed instructions for the building of the organism, whether it is a one-celled bacteria, a giant oak tree, or a human being. The full set of DNA present in an individual organism is called its genome. So Dr. Folks, if I took all of my DNA, I could have Dr. Folks' genome. If you took all of your DNA, you would have your entire genome. Okay, it's your complete set of DNA. And it's found in the nucleus of the cell. So remember that. It's found in the nucleus of the cell, and it's found in every single cell. So, interesting fact, if you took all of the DNA out of just one cell in your body, how much DNA do you think that would be? Like if I spread that DNA out and tried to figure out its length. The answer is 6 feet. So one cell is microscopic, you cannot see it. However, within the nucleus of that one cell, it's holding 6 feet worth of DNA, which is crazy. And if you took all of the DNA and all of your cells combined, you could go back and forth to the sun. I think it's like 600 million times or something ridiculous like that. In eukaryotes, including humans, all of the genome is laid out in these long linear strands of DNA. So rather than having the genome in one super long DNA strand, eukaryotic DNA exists in numerous smaller, more manageable pieces called chromosomes. So those are shown there as well. And then, of course, within chromosomes, you have lots and lots of genes, which then go on to produce proteins. So the definition of gene would be a sequence of bases in a DNA molecule that carries the information necessary for producing a functional product, usually a protein molecule. It can also be an RNA, but we won't talk about that. So just remember that it's a protein. So within the long sequence of bases of your entire DNA, there are relatively short sequences. And on average, those are about 3,000 bases long, so 3,000 letters in an actual gene. Okay, so now we talked about your DNA, and then we talked about an entire genome, and then we talked about individual genes on chromosomes, and now we need to talk about alleles, so another vocabulary word to know. Within a species, individuals sometimes have slightly different instruction sets for a given protein, right? So maybe hair color. Our hair is made of protein. So my hair color is a light brown. My instructions say that, so there's an allele that says make brown hair, but yours may be different. These instructions can result in a different version of the same characteristic. These alternative versions of a gene that code for the same feature but slightly different variation are called alleles. And you could think of them as functioning like alternative recipes for chocolate chip cookies. So my recipe for chocolate chip cookies and your recipe for chocolate chip cookies are going to be slightly different. Just like my recipe for my hair and your recipe for your hair is going to be slightly different. And that means that we have different alleles. So it's different versions of a gene that code for the same trait. A trait is a physical characteristic. All right, so go ahead and answer this question. Pause the video if you want more time. Melanin is a protein, is the answer, responsible for skin pigment. People who have albinism carry a mutant allele that does not make melanin. All right, so here's the take-home message. You can read that on your own time. Something I didn't mention yet is that in prokaryotes, the DNA occurs in circular pieces. So eukaryotes, us, we have chromosomes, which are those linear chunks. But prokaryotes have one circle of DNA. They have a lot less DNA than us. So they can compact it into one circle and be okay. We can't really pull that off. If we compare the amount of DNA present in various species, we will see a paradox. There does not appear to be any relationship between the size of an organism's genome and the organism's complexity. So what this chart is showing you is genome size of various organisms. So remember, a genome would be all the DNA. So my genome size in millions of base pairs as a human is 3.4 million base pairs. But an onion, which I'm way more complex than, an onion has 18 million base pairs. That's 5 times as much DNA as a human. And then a salamander has more, and then an amoeba, which is a single-celled organism, has even more. It has over 600 million. What's that all about? Because it's debatable whether we're the most complex species on the planet, but surely we must be more complex than an onion. But we're not if you measure complexity by the amount of DNA an organism has. An onion has more than 5 times as much as a human. We don't fare any better compared to some other seemingly simple creatures either. Like that salamander species has about 25 times as much DNA as we do. So why does an onion have more DNA than us if DNA amount, if genome size that is, does not relate to the complexity of the organism? That's because it is the proportion of DNA that codes for genes matters, and this also has a lot to do with evolution. We'll get to that later. So older species are less complex, but they have had more time to accumulate extra DNA that isn't necessarily coding for genes. Okay, so remember, genes are the things that actually form recipes for making proteins to do things. But your DNA is not all made up of genes. In fact, the majority of humans' DNA is not made up of genes. In humans, genes make up less than 5% of the total DNA. In many species, the proportion of the DNA that codes for genes is even smaller. In virtually all eukaryotic species, the amount of DNA present far exceeds the amount necessary to code for all of the proteins present in the organism. The fact is, a huge proportion of base sequences in DNA do not code for anything and has no obvious purpose. Some biologists call it junk DNA. I don't like calling it junk DNA because more and more we're learning that these non-gene DNA sequences are actually very, very important a lot of the time. So I don't really like calling it junk DNA. It's non-coding DNA. It does not code for genes. Bacteria and viruses tend to have very little non-coding DNA, with genes making up 90% or more of their DNA. So E. coli is a bacteria. You can see that 90% of the coding DNA, of the DNA found there, is coding for genes. So here I'm showing you coding versus non-coding regions of DNA. So we're within the cell, we went in the nucleus, we are now within the whole genome, zoomed in on one chromosome, on a chunk of it. We're looking at that DNA. You can see two different genes here. They're in the gray box. And you can see all this DNA in between those two genes. If it's not directly coding for proteins, what is it doing? Well, it's not junk, it does important stuff, but you don't need to know what it does. So just know it's non-coding regions. And that 75% of DNA does not code at all. And then even within genes we have non-coding regions called introns, but you don't need to know that either. Just know that you have non-coding regions between DNA. So which species has the most DNA that does not code for genes? Alright, the answer would be humans in this example. Because only 2% is coding for genes, that means 98% of our DNA is non-coding. So only a small fraction of the DNA in eukaryotic species is in genes that code for protein. The function of the rest is still being looked at. It's a big area of research. So how do genes actually work? Every cell contains all of the information needed to manufacture every protein in the body, but having the instructions is not the same as having the products. The genes and strands of DNA are a storehouse of information, an instruction book, if you will. But they are only part of the process by which an organism is built. If the genes of an organism carry a trait, its genotype, that is like the recipe in a cookbook. Then the physical manifestation of the instructions, the organism's phenotype, are the cookies. Okay, so the genotype is going to be that DNA recipe for your cookies. The phenotype would be the actual cookies, the physical manifestation of the instructions. More examples. I have a genotype, a DNA recipe of gene for my hair color. That's the phenotype. Okay, I have a genotype, different genes, DNA recipe for my phenotype, blood type. I have a genotype, that DNA recipe for the phenotype that is if I am male or female. How does a gene, a sequence of 4 letters of 4 bases within a section of DNA, how does it affect a flower's color, or the shape of a nose, or the texture of a dog's fur? How does it affect all of those phenotypes? Well, the process occurs in 2 main steps. I want you to know that. There's transcription and there's translation. Transcription is when a copy of a gene's base sequence is made. Okay, so that's copying. Just like if something is transcribed, that just means you made a copy of it. Translation is when that copy is used to direct the production of a protein. So if I was to translate English into Chinese, those are completely different languages. Translation within the cell is the same thing. You're translating from nucleotides to a completely different language of proteins. I like figure 5-11. It's an overview of the steps from the gene to the protein. So in transcription, which occurs in the nucleus in eukaryotes, the gene's base sequence or code is copied into a middleman molecule called mRNA. So transcription is when you go from DNA to mRNA in the nucleus of the cell. You need to know that. And the analogy here would be the DNA is your recipe cookbook. And then the mRNA would be copying that recipe cookbook. Maybe I'm going to write it down on an index card, take it home so I can have it forever. Alright, so that mRNA, which is the recipe card that I copied, is going to leave the nucleus and go out to the cytoplasm. And then it is in the cytoplasm of the cell where that mRNA message is used to build proteins through translation at the ribosome. So you need to know that. Alright, so we translate that recipe into ingredients that we combine, and then we make our chocolate chip cookies or our protein molecule. Okay, so again, DNA stored in the nucleus. It's transcribed, copied into mRNA in the nucleus. mRNA leaves the nucleus, goes to the cytoplasm. In the cytoplasm, it gets to the ribosomes and is translated into proteins. Okay? Another thing, it's called transcription, like just copying, because you're using nucleotides between DNA and mRNA. They both contain nucleotides. But translating something means a different language, and nucleotides are used for mRNA, but amino acids, which would be a different language, amino acids are what make up protein molecules. Okay, so pause if you need more time. The sequence of nucleotides in the DNA determines the genotype of an organism, whereas the physical manifestation of the DNA code is called the phenotype. Alright, so the sequence of nucleotides is the genotype, and then the physical characteristic is the phenotype. Hopefully I said those right. So genotype was the first one, and then phenotype was the second one. They sound similar, so sometimes I say them wrong. Alright. So transcription and translation kind of freak people out, and they get them switch swapped all the time. Sorry. And the way that I personally remember them is I think about what the word means in English. So again, to transcribe just means to copy. So you're going from English letters to a copy of English letters. So you're going from nucleotides in a DNA to a copy of nucleotides in an mRNA. Translation is, you're going from one type of word to a completely different word because you're in a totally different language. So you're going from the language of nucleotides in the mRNA to the language of amino acids in the proteins. Transcription has a lot of steps. I don't really need you to know all the steps. I just need you to know that DNA is unwound. Those rungs that are bound together by those hydrogen bonds are fairly easy to separate. So they unwind the DNA, separate those the complement strands of base pairs, and then an enzyme called RNA polymerase will go through and make the mRNA transcript to base pair with that DNA sequence. And then that mRNA goes off, it's processed. There's a lot of processing steps, they're very important, but we don't really have time to go into all of that detail. And then in translation, the mRNA copy of the information from DNA is used to build functional molecules of protein. For translation to occur, you must first have a large number of the amino acids floating around, because remember, amino acids are put together to make a protein. You need to have these ribosomal subunits, so you need to make a ribosome, which actually comes in two different parts, but just know that you need ribosomes. And then finally, you need a molecule that can read and translate the mRNA code from base sequence into a protein. So that's called a transfer RNA or a tRNA. Those are the interpreters, if you will, of the mRNA code that link specific bases on the mRNA with specific amino acids. This is translation in more detail. There it's showing you the tRNA, which is your interpreter for translation. On the bottom is your mRNA, and tRNA reads these codons, which are 3 base pairs, and it says, oh, this is ACG. That means that I need to add a threonine amino acid. You don't need to know which codons equal which amino acids. I just want you to know that the interpreter is the tRNA, and it can translate that message. And that it brings in the amino acids as well. These are all the different possible codon combinations. So remember, it's those 3 nucleotides added up together. You're seeing a U instead of a T here because in RNA we don't have thymine, which is T, we have uracil, which is U, but it's the same difference for all intents and purposes. And just know that that triplet code, that codon makes says which amino acid needs to be added. Okay, so some amino acids have multiple codons that code for them, because you only have 20 amino acids, but you have 64 possible codon combinations, so there's redundancy in the code. How do we regulate genes? So we have this entire genome, but again, remember, my brain cell, my liver cell, they look different, they have different jobs. What chunks of the genome are they actually transcribing and translating at any given time? They're not going to use the entire genome. They're going to use the parts that they need to be a brain cell or a liver cell or whatever type of cell it is. So how do we flip on or off transcription? How do we say turn this section of DNA genes on so I can be a liver cell, or turn these off so I can be a brain cell, etc.? This is very important. Practical example is that gene expression that's turning on and off your genes. Gene expression can be linked with specific patterns of phenotypes. The example here was that impaired transcription, impaired meaning decreased, so impaired transcription, less transcription of some genes was associated with an effective stress response. At the same time, there was an increase in transcription in genes associated with an inflammatory process. But that's an example in your book. Mutations. Sometimes something occurs to alter the sequence of bases in an organism's DNA. And that alteration is called a mutation. So you need to know that. And it can lead to changes in the structure and function of the proteins produced. Remember we talked about proteins as biomolecules. We talked about how their structure is so important, and denaturing that structure can completely mess up the protein. It can't do its job anymore. If you don't remember that, you need to go back to that. That's chapter 2. So mutations can kind of act like denaturing. It can completely change the shape of the protein and make it not functional at all. So sometimes mutations result in serious, even deadly problems. Most of the time, though, they don't really have any effect. And sometimes, it's very rarely, but sometimes they can be beneficial. And that is hugely important for the theory of evolution. So damage to the genetic code has a variety of causes and effects. See here, all these different mutations are causing different things to happen in these fruits and vegetables. That could be bad. It could be good, though, if it completely changed the flavor of the vegetable and made it taste better, right? It could be beneficial. An example of a bad mutation would be breast cancer. So when two human genes called BRCA1 and BRCA2 are functioning properly, they help to prevent breast cancer by keeping cells from dividing uncontrollably. But if the DNA sequence of either BRCA1 or BRCA2 genes is altered through mutation, then the gene's normal function is lost. When it's lost, then the person carrying the mutated gene has significant increased risk of developing breast cancer. Why is that? Because cancer is the uncontrolled division of cells. That's why you get tumors. Those are masses of cells that kept dividing and dividing and dividing and dividing when they weren't supposed to. Currently, more than 200 different changes in the DNA sequences of these two genes have been detected in breast cancer patients, and each of those changes or mutations results in an increased risk of developing breast cancer. So, true or false, mutations are never beneficial? The answer is false. They can be. They can also be disastrous, but most of the time they're neutral. Here's another example of a mutation. We use fruit flies, which you've probably seen them. They're really tiny. They look like gnats in your kitchen sometimes, especially if you leave out like bananas too long, for example. But these little fruit flies, we use them in the lab. We go in and we look at their DNA, like we change genes and mutate genes and see what the phenotype, the physical effect is of those mutations. So here, the mutation that has been introduced to the fruit fly to make the mutant can change the protein needed to make the eye shape. So we get almost a complete loss of the eye shape. But a lot of the times, the mutations aren't going to change the eye shape. You'd still have a normal looking eye. I want you to remember that the paradoxical fact about mutations is that they are essential to evolution. So they're not just bad. Like, we need mutations to some extent. Because the mutations that don't kill an organism or reduce its ability to survive and reproduce can be beneficial. Every genetic feature in every organism was initially the result of a mutation. So the fact that humans are the way they are compared to a bacteria, we evolved from a bacteria because we had different mutations that were beneficial to us. And then they kept accumulating and accumulating until we evolved eventually into a human being. So they are important and beneficial. So why is it that dentists put that heavy apron over you when they x-ray your teeth? That's because ionizing radiation, such as x-rays, is radiation with enough energy to disrupt atomic structure. That would include the atoms in your chromosomes, so it can break apart chromosomes by removing the electrons. But ionizing radiation, like x-rays, cannot pass through lead. So the lead apron a doctor or dentist puts over you when you get an x-ray protects your body from the ionizing radiation. We don't want the ionizing radiation to get into our body, get to the chromosomes. It could mess them up. It could cause a lot of mutations. Okay, so sometimes mutations are bad, like in the case of cancer. So we don't want more than necessary mutations. Mutations are alterations in a single base or change in a large segment of DNA. They could include a big chunk of DNA. Typically, it's a single base, though. Like an A would become a C or something. They're rare, but when they do occur, they can disrupt normal functioning of the body, although many are neutral. Extremely rarely, mutations may have a beneficial effect. In the next section, we examine how even tiny changes in the base pair sequence of DNA can lead to errors in protein production and profound health problems. So how can people respond so differently to alcohol? So here's an example. Isabella joins her friends in sipping wine during a dinner party. As the meal progresses, her companions become tipsy. Their conversations turn racy, their moods relax. They refill their glasses, reveling in a little buzz. Not so for Isabella. Before her first glass is empty, she experiences a fast flush response, which means her face turns red, her heart begins to race, and her head starts to pound. Worse still, she soon feels the need to vomit. So how come Isabella feels so much different when she drinks alcohol compared to all of her friends? Well, it comes down to a single difference in a single base in her DNA. A single mutation can influence dramatically a person's behavior, digestion, respiration, and general ability to function. The single base pair change leads to the production of a non-functioning enzyme, so a messed up protein that can't do its job, and the lack of a function leads to physical illness. Poor Isabella. So when we consume alcohol, our bodies start a two-step process in which the alcohol molecules are converted from their intoxicating form into innocuous molecules. Each of the two steps is made possible by a specific enzyme whose assembly instructions are coded in the DNA of most people. Fast flushers like Isabella, so people that turn red, they flush red, they complete the first step of breaking down alcohol, but they cannot complete the breakdown because they carry a defective gene instruction for making the protein aldehyde dehydrogenase. Kind of a big funny word, but that's an enzyme that makes it possible to do the second step of the alcohol breakdown process. Because she cannot break down the alcohol, a poisonous substance accumulates, and we get the fast flush reaction due to the toxic side effects of that substance. So although the details differ from case to case, the overall picture is the same when it comes to many, if not most, inherited diseases. Okay, so we're just using this example with alcohol to show you how inherited diseases work in general. The pathway from mutation to illness includes just 4 short steps. First, a mutated gene codes for a non-functioning protein, commonly an enzyme. Next, the non-functioning enzyme cannot catalyze the reaction as it normally would, bringing it to a halt. Okay, so it cannot perform its job. Next, the molecule with which the enzyme would have reacted accumulates. Think of a blocked assembly line. So you get a buildup of all that stuff, and then the accumulating chemical causes sickness and or death. So if you remember Tay-Sachs disease, remember what that was? It had to do with lysosomes. Go back if you don't remember. But Tay-Sachs is an inherited disease. It's got horrible consequences. A protein, an enzyme, has a mutation in its DNA code. So you get a non-functioning enzyme that is supposed to break down lipids within the lysosome. But because it doesn't, we see an accumulation of lipids. The accumulation of lipids busts open the lysosome, spilling out all of that acidic juice, which will then kill the entire cell. So you can see those 4 steps in the case of Tay-Sachs as well. Go ahead and pause this if you need more time. A mutation occurs in the DNA of a gene, because the gene is always going to be DNA. But the disease is caused by translation of a non-functional protein. Go ahead and do this one too. The answer is number 5. So number 1 would be a point mutation. Can you find what's different in number 1? What's different is you see the tag that has become a TAT in number 1. So that G became a T. So that's a point mutation. That's when a single nucleotide gets messed up. Number 2, can you see what's different? Right. So at the end, we're missing a G. That's a single nucleotide deletion. Number 3 is a large deletion. We're missing a whole lot of letters. Number 4, we actually have extra letters, so we call that an insertion mutation. So an example of a deletion in the sequence is number 2 and 3. So number 5 was the right answer. So you can have a tiny deletion or you can have a huge deletion. Okay, go ahead and read that. Understanding a phenomenon is nice, but controlling it takes this satisfaction to entirely new levels. This may explain why there is so much excitement surrounding the field of biotechnology. That's when technology is used to modify organisms, cells, and their molecules to achieve practical benefits. The modern emphasis in biotechnology is on genetic engineering. And genetic engineering is defined as the manipulation of an organism's genetic material by adding, deleting, or transplanting genes from one organism to another for useful purposes. A practical application of this would be corn. So for thousands of years, humans have been practicing a crude and slow form of genetic engineering, which is the manipulation of a species' genome in ways that do not normally occur in nature. In its simplest form, genetic engineering is the careful selection of the plants or animals used as the breeders for a crop or animal population. Through this process, farmers and ranchers have produced meatier turkeys, bigger chickens, seedless watermelons, that's not natural, and big juicy corn kernels, like in this figure. So the ancestor of corn did not have these giant delicious kernels on it. It looked like that ancestral corn in that picture. Same thing with strawberries. If you go find wild strawberries, they're very tiny. They do not look huge like the ones you buy in the store. I also used to buy free-range chickens. Those are much smaller than those chickens you would buy mass-produced in the store. But what used to take many generations of breeding can now be accomplished in a fraction of the time using recombinant DNA technology. All that means is that we combine DNA from 2 or more sources into a product. So there's vast benefits to doing this genetic engineering and using recombinant DNA. Almost 10% of the world's population suffers from vitamin A deficiency, and vitamin A deficiency can cause blindness, and it does cause blindness in 250,000 children each year. And these nutritional problems are especially severe in places such as Southern Asia and Sub-Saharan Africa, where rice is a staple of most diets. Addressing this global health issue, researchers have developed what may be the model for solving problems with biotechnology, and involves the creation of a new crop called golden rice. Mammals generally make vitamin A from beta-carotene, which is a substance found in abundance in most plants. It's what makes carrots orange, for example. Beta-carotene also is found in rice plants, but not in the edible part of the rice grains. So what we eat in rice would not have beta-carotene in it. Researchers set out to change this by inserting into the rice genome from other species 3 genes that code for the enzymes, proteins, used in the production of beta-carotene. Okay, so they took genes from daffodils and bacteria and stuck them into rice so that the rice can now make beta-carotene, so that people eating the rice can get the beta-carotene they need in their diet, and then they can get vitamin A and not have vitamin A deficiency anymore. You might say, well, why don't they just eat more carrots? Well, we're talking about areas with a lot of poverty where their staple diet is rice, and basically only rice. They do not have abundant fruits and vegetables to eat. They're only eating rice, so that's why they're malnourished in vitamin A in the first place. It's clear that the transplanted genes are working because the normally white rice takes on this golden color from the accumulated beta-carotene. The rice doesn't yet supply a full day's requirement of vitamin A in one serving, but it does provide a significant amount. It was first developed in 1999, but new lines have been produced with more than 25 times as much vitamin A than the original strains. Field tests of golden rice are still underway, but is viewed as one of the most promising applications yet of biotechnology. Currently, more than 170 million acres worldwide are planted with genetically modified crops, most containing built-in insecticides and herbicide resistance. Okay, so a lot of people have problems with all of the insecticides sprayed on their produce, which makes sense. A way to get around that is that they went in and genetically modified plants to be more resistant to insects. So if you go and you buy non-GMO produce from the grocery store, the chances that it's got insecticides is much greater than if you bought the GMO produce. So that's a huge downside to the non-GMO movement. It requires a lot more insecticide to keep those fruits and vegetables looking healthy and not getting eaten up by bugs. So 86% of all corn grown in the United States is genetically modified. 93% of all cotton is genetically modified. 93% of all soybeans are genetically modified. And that's because many plants have had insecticides engineered into them, and many plants also have herbicide resistance genes engineered into them. And the reason for the latter is that it reduces the amount of plowing required around crops to remove weeds. So they're going to be more resistant to bugs and more resistant to weeds. So I know, like, non-GMO has become a big thing. People are like scared of GMOs, but we've been doing this for a long time. And a lot of research goes into this because it has a lot of benefits. And I think I already really explained that. This is just a practical example showing that insect resistance, how we took a gene out of a bacteria that would help make the plant cell resistant. So you can go over that in your own time. So what proportion of corn grown in the U.S. is genetically modified? You can look at that picture if you don't remember the percentages I told you. It's kind of also testing your ability to read a chart. And the answer would be number 2, 86%. So you can see in the pie chart for corn, the blue is the proportion of crops that are genetically modified. So that's 86%. Okay, so what is your opinion? I can't get your actual answer. I use this question in my lecture courses just to kind of have a discussion about if benefits outweigh the risk of GMOs. So think about that. This could be something that you talk about on the discussion forum for this week. Biotechnology is not just important for agriculture, it has the potential for improving human health and also the criminal justice system. We won't go into that last part, but that's the last section in your book for the chapter. One example is the treatment of diabetes. So diabetes is a chronic disease in which the body cannot produce the chemical insulin. You need insulin to break down sugar in your blood. Complications from diabetes include vascular disease, kidney damage, and nerve damage. As recently as 1980, if you were one of the approximately 15 million Americans with diabetes, each day you would treat the disease by injecting yourself with insulin extracted from the pancreas of cattle or pigs that had been killed for meat. For most diabetics, the insulin injections kept the disease under control, but the traditional process of collecting insulin this way was difficult and costly. So we could not make insulin for our own bodies, so we would harvest it from animals and then inject it into ourselves so that we could break down the sugar in our blood. But in 1982, everything changed when a 29-year-old entrepreneur, Bob Swanson, joined scientist Herbert Bauer to transform the potential of recombinant DNA technology. Remember we talked about recombinant DNA technology. What they did is they started the biotech revolution pretty much. They worked with scientist Stanley Cohen, and the three of them used enzymes to cut out the human DNA sequence that codes for the production of insulin. And they then took that human gene to make insulin and put it into a bacteria, E. coli, to create a transgenic organism. That transgenic organism could then pump out a bunch of human insulin. So now people can inject themselves with synthetic human insulin from a genetically modified bacteria. And this has saved millions of dollars and helped millions of people. And it wasn't just important for diabetes. This had huge effects in research in general. This technology became so important. Today, more than 1,500 companies work in biotechnology industry, and their products generate more than $40 billion revenue each year. Oh, sorry. The last section is cloning. You can clone things like genes, whole cells, organs, even whole animals. It has pluses and minuses. Part of your homework is a video looking at cloning organs for organ transplants. So definitely check that out. That's interesting. Are there any medical justifications for cloning? At what point is it okay or not okay? Is it okay to clone cells? Is that justified? Is it justified for organs, animals, humans? Medical researchers see much to gain from cloning. In particular, transgenic animals containing human genes can be very valuable. But then ethics come into play when we start talking about entire humans. Should we even pursue that? There is near unanimous agreement among scientists that human cloning to produce children should never be attempted. Some of the reasons cited relate to problems of safety for the mother and the child, legal and philosophical issues relating to the inability of cloned individuals to give consent, problems of the exploitation of women, and concerns regarding identity and individuality. So governments are struggling to develop wise regulations for this whole new world of cloning. It's just another opinion question. You know, how far can we go with cloning? How far should we go? All right, chapter 6. I forgot to put this on the slide, sorry. The sections covered will be 6.2 to 6.4. Alright, so that would include 6.3. And then 6.6 through 6.9. So that would include 6.7 and 6.8. And then 6.11 through 6.12, 6.14, and 6.17. We will talk about cell division, the different types, mitosis, and how it takes worn out old cells and replaces them with new duplicates. We will talk about meiosis and sperm and egg cells. How are they made? We'll describe the sex differences in the chromosomes and consequences when we have changes in the normal chromosome number. This is just showing you cell division. So you're going from one cell to two here, and this is an example of meiosis. 6.2 is just revisiting the fact that prokaryotes on the left have one large circular piece of DNA, whereas eukaryotic cells on the right have those chromosomes. And those chromosomes are made up of chromatin. Chromatin is DNA wound around these histone proteins. And the reason why we do that is to make sure we can pack all that 6 feet of DNA into the cell. That's all I want you to know from that section. The cell cycle has phases, and the cell spends long periods of time occupied with activities related solely to the growth of the cell, and then they may suspend those activities as they segue into a period devoted exclusively to reproducing the cell. Before we continue discussing the cell cycle, we need to note an important distinction among the cells of the body. All the cells of a multicellular eukaryotic organism can be divided into two types, somatic and reproductive. Somatic are the cells forming the body of the organism. So the root of that word, soma, means body, so body cells. And then reproductive cells are the sex cells. That is the gametes, which are sperm and egg, that give rise to humans when we combine those two gametes. So somatic cells and reproductive cells are use different methods of producing new cells. Okay, so somatic cells are going to use mitosis, and then reproductive cells are going to use meiosis. Unfortunately, they sound similar, so you'll have to work on trying to memorize that. Alright, you don't need to know all these details. I just want to show you this is the cell cycle. In purple is interphase, and then in blue is mitosis. 90% of the cell cycle is actually spent in interphase. This is when the cell is getting ready for cell division. It's going to replicate all of the DNA in interphase, and it's going to replicate all of the organelles. It's also going to grow in size so that when it splits into two cells, you'll have two normal sized cells, not two really tiny cells. Okay, and then 10% of the whole cell cycle is mitosis, and that's the actual dividing to make two genetically identical cells. You don't need to know all the phases of the cell cycle. I want you to know interphase versus mitosis, but I want you to know that there are cell cycle checkpoints. This is like a control system. There are 3 of them. They are critical points in the cell cycle at which progress is blocked until specific signals trigger continuation. That's because if we have a problem replicating the DNA or something like that, we don't want to then go through with mitosis. You could get completely messed up numbers of chromosomes in those cells or huge numbers of mutations if you did not replicate the DNA properly. So that is the purpose of these cell cycle checkpoints. In the case of cancer, what the cancer cells do is they just speed through all of these checkpoints. They just drive right through that bar. They don't even care. And so a lot of mutations accumulate in these cancer cells as they keep dividing, way more than they should, even if they're messed up. And that can lead to worse and worse and worse cancer. All right, interphase and mitosis. Checkpoints. Before we can have cell division and interphase, we get that chromosome replication. All right, so this is just showing you the parent DNA molecule. You split that double strand apart, and then each of those individual strands will become a complementary template strand to then get two identical daughter DNA molecules. So that's DNA replication. So remember, A base pairs to T and G to C. You don't need to know all the detail. I just want you to know the process of DNA replication occurs in two steps. The first is unwinding and separation of the two strands, and then the second is reconstructing and elongating the new complementary strand. The daughter molecules are very identical, except there is some error. So it's very low, but it's not a zero rate of error when we are replicating all of those DNA base pairs. A variety of mutations can occur during replication when DNA is damaged because the protein that actually puts in the complementary base pair during replication can sometimes mess up. Just like if you were taking a piece, okay, let's say you have a page from a book, you rip it out, and you're supposed to transcribe that entire page over. Well, you might occasionally miss a word or a letter or something like that. Okay, that would be like an error in DNA replication. And for that reason, new genes can sometimes enter a population and be acted on by evolution. So again, we're just talking about those mutations again. They can be beneficial, usually neutral, sometimes bad. So this is where those mutations mostly arise from. Now you know. Okay, so let's say we just did DNA replication. We have the complementary strands here, but there was an error. So for which one of these did an error occur during that DNA replication? This is also testing your ability to remember what base pairs with what. So the answer would be number 1. So G should base pair to C and T should base pair to A. But we see here, number 1, if we go 4 letters in from the left, that A, its base pair is a G. Well, that's incorrect. That should be a T. Alright, so every time a cell divides, it must first duplicate its DNA. Replication is how we duplicate DNA. And it results in 2 double-stranded DNA molecules that are virtually the same genetic information, although some errors do happen. Alright, now we're going to talk about mitosis and meiosis, more specifically. So mitosis, its purpose is to replace worn-out old cells with fresh new duplicates. Its purpose is also for us to grow larger, right? So there's going to be a lot of mitosis taking place in a little infant becoming an adult. What I want you to remember in red is that mitosis makes 2 genetically identical daughter cells from one parent cell. Alright, so in my body right now there's a skin cell that is undergoing mitosis to make 2 identical cells. So for mitosis to begin, the parent cell replicates its DNA, creating a duplicate copy of each chromosome. Once this task is completed, mitosis can take place. Mitosis occurs in 4 steps in which the now duplicated chromosomes are separated into 2 identical sets in 2 separate nuclei, after which the cytoplasm and the rest of the cell are divided into 2 cells that pinch apart. So here you're seeing the pinching apart of 2 identical daughter cells. Before mitosis begins, the chromosomes replicate, becoming two identical linear DNA molecules. They are held together in a region called the centromere. I don't really need you to know that, but I need you to know about the identical replication of the chromosomes. I'm not going to go through all the steps of mitosis. I'm going to breeze through them. Remember that the daughter cells are genetically identical. Interphase, we talked about that. That's when the chromosomes are replicating, cells getting bigger, organelles are duplicating. So know that. What's happening with these chromosomes here? Here they're being separated. The result of mitosis is two genetically identical cells. You must know that for sure. All right. Cell division out of control may result in cancer. So I kind of talked about that. So has cancer affected anyone you know? This is something I ask my students. It's something you can discuss in the forum this week. Cancer is defined as an unrestrained cell growth and division. It can damage tissues. It can metastasize, which means that the tumor can get up and travel through your body to other parts. So I used to research breast cancer, and breast cancer has a tendency to metastasize to bones. And we were interested in why the bone. Like, why do breast cancer cells want to travel to the bone specifically? And so different types of cancers are going to metastasize to different regions. And this is the second leading cause of death in the United States. Alright, so this is showing you on the left we have a plate, a petri dish full of cells. So normal cells will divide and kind of just fill up the plate and then they'll stop dividing. But cancer cells, they don't care that they've already filled up their space. They're going to keep dividing. They're going to fly through those cell cycle checkpoints that they're supposed to stop at, and they're going to form a tumor. We can have benign tumors and malignant tumors. So benign tumors are just going to be a mass of tissue. They're not necessarily going to spread through the body and keep forming tumors in other locations, but malignant tumors will metastasize and cause some serious problems throughout the entire body. How does cancer actually kill the organism? Somewhat surprisingly, it's not because of some chemicals or genetic property of the cancer cells themselves. It's simpler than that. So as a tumor gets larger, it takes up more and more space and presses against neighboring cells and tissues. And eventually, the tumor may block other cells and tissues from carrying out their normal functions and even kill them. This cell dysfunction or cell death can have disastrous consequences when the affected normal tissue controls processes critical to life, such as breathing, heart function, or the detoxification process in the liver. This is why pancreatic cancer is so fatal. So why do we do chemotherapy and radiation? Like, what's that all about? Well, to treat cancer, the rapidly dividing cells must be removed surgically or killed, or their division at least slowed down. So currently the way we try and treat cancer is killing and slowing down the rapidly dividing cells using chemotherapy and by radiation. So in chemotherapy, drugs are administered that interfere with cell division. That's going to slow down the growth of tumors. And then like chemotherapy, we have radiation, which works by disrupting cell division. But unlike the drugs used in chemotherapy, which circulate throughout the entire body, radiation therapy directs high energy radiation only at parts of the body where a tumor is located. This is why your hair falls out and you have digestive issues if you undergo chemotherapy or radiation, because your hair cells and your digestive cells are rapidly turning over. So you're like making new cells all the time because you need them. Whereas like your bones don't necessarily make new bone all the time. They kind of stay where they're at for quite some time. So the cells in your body that are rapidly turning over are going to also be affected by chemotherapy and radiation, because you're trying to basically kill the cancer cells before you kill your normal cells, because your normal cells are always dividing too, but the cancer cells are always dividing even more, so you're going to kill those off hopefully. But any cells that are rapidly dividing in your body are going to be killed as well, unfortunately. All right, which statement is false about a malignant tumor? You probably want to pause this to read all of these. The answer is number 2. I don't think I actually went into apoptosis. You need to look that up in your book. It's programmed cell death. Cancer cells are full of mutations and they are breezing through those checkpoints, for example. So normally when a cell has a lot of mutations accumulated or the cell is not stopping when it should during those cell cycle checkpoints, apoptosis will occur so that it can kill that bad cell and not keep replicating it through mitosis. But cancer cells are like, no, you can't kill me, and they just keep dividing anyway by avoiding apoptosis. This one you don't really need to know. So cancer is unrestrained cell growth and cell division. It results from mutations to genes important in controlling the cell cycle, specifically those checkpoints. And the treatments for it focuses on killing or slowing down the fast-growing and divided cells. Meiosis. So meiosis does introduce genetic variation. So when we talked about mitosis, I said there are 2 genetically identical daughter cells. Meiosis is going to make sperm and egg cells that are not genetically identical. They're going to have genetic variation. There are 2 ways in which an organism can reproduce. Many organisms like bacteria and fungus and even some plants undergo asexual reproduction. So asexual reproduction, that is when a single parent produces an identical offspring. But other organisms, including most animals and plants, undergo sexual reproduction. That's when offspring are produced by the fusion of 2 reproductive cells in the process of fertilization. And some species, particularly among plants, can use both methods. Alright, so humans use sexual reproduction, and a benefit of that is because a combination of DNA from 2 separate individuals is passed on to offspring, so that the resulting offspring are genetically different from their parents and from one another. So these are terms you need to know. Gametes are going to be the sex cells. These are going to be your sperm and egg. Diploid's going to mean that it has a normal chromosome number. So we have 46 chromosomes as humans. We have 2 sets of 23 though. Okay, so I get 23 chromosomes from my mom, and I get 23 of the same type of chromosomes. They're not identical, but the same type of chromosome from my dad. Alright, so 23 from my mom plus 23 from my dad equals 46. So diploid, di, 2, means there's 2 sets of chromosomes in my body, in my cells. Haploid is going to be half the number, right? So my egg cells and then also sperm cells are going to have half the number of chromosomes, okay? So they only have 23 chromosomes. The reason for that is to maintain a stable genome size. So if my egg cells didn't have 23, if they had 46, when I have a fertilized egg, then that would have way too many chromosomes in it. It would actually have 96. We don't want that, and that's why we have half the number in our sex cells. So at fertilization, 2 haploid cells, each with 1 set of 23 chromosomes, merge and create a new individual with the proper diploid human genome of 46. When the time comes to reproduce, this new individual through meiosis also will produce haploid gametes that have only a single set of 23 chromosomes. With sexual reproduction then, diploid organisms produce haploid gametes that fuse at fertilization to restore the diploid state. In this way, meiosis maintains a stable genome size in a species. You absolutely need to know that. Okay? Meiosis achieves more than just a reduction in the amount of genetic material in the gametes. It is important for genetic diversity because you have 2 copies. Remember we talked about alleles? This is why we have 2 alleles of every gene. So do 2 different variants of every gene, right? So my mom has brown hair, so I got an allele for the brown hair, but then my dad has blonde hair, so I got an allele for the blonde hair, all right? So those are 2 different variations of the same gene, which encodes for hair protein. Right, so this is JFK, of course, on the right. So these are the Kennedy brothers. They have some physical similarities, but they obviously look different from each other. And that's because each egg or sperm is produced with a varied combination of maternal and paternal alleles, so that each of the gametes an individual makes carries a unique set of alleles. As a consequence, each offspring inherits a slightly different assemblage of each parent's alleles. All offspring resemble the parents, yet none resembles them in exactly the same way. They are not clones. This variation among offspring can be seen among the brothers in the photo that opened in the chapter. So if you remember the Jackson 5 picture. Alright, so here's the 2 important outcomes in red that you should absolutely know. Alright, so meiosis results in the formation of genetically non-identical gametes. Okay, so remember, there are 2 different types of cells, somatic and the reproductive ones. So those are going to be meiosis, but those somatic ones are going to be mitosis. So remember that from the beginning. Alright, so your gonad cell is going to make the sperm or the egg. I apologize. So the gonad cell is not the sperm or egg. I apologize. The gonad cell is the parent cell that's going to make the sperm and the egg. So the gonad cell is diploid, and then it goes through the 2 rounds of cell division to make these haploid daughter cells. Those are the sperm and egg. And then they're all genetically different. Okay, so the outcome: 4 haploid daughter cells, just 1 set of chromosomes, unique combination of traits, unique combination of alleles. This is the entire meiosis process. All I want you to know here is that there's 2 rounds of division. Okay, so it kind of looks like mitosis, but there's 2 rounds of it, and in the second round you see how we end up with 4 cells instead of 2. And it's because there's 2 rounds of division that the cells end up haploid in meiosis instead of diploid like they did in mitosis. Okay, so the result is 4 genetically non-identical cells. Meiosis occurs only in the gamete-producing cells, not in the somatic ones. It is preceded by DNA replication. It consists of 2 rounds of cell division, and the final product is 4 haploid gametes. Male and female gametes are produced in slightly different ways. I'm not really going to go into how sperm versus eggs arise from their respective sex or gonad cells, but they are both haploid, and I want you to know why they are haploid. I've been talking about it. Think about it. Go back and review it. Remember that meiosis has genetic variation introduced, and that's because we have these crossing over events. So the chromosomes are going to swap pieces of DNA with each other, and that's an important source of variation. That's shown here. So you have the homologous chromosomes swapping some material here, and then you end up with these chromatids that all look different from each other. So crossing over doesn't create new alleles, but it does create new combinations of alleles. I want you to know that part. Okay, so it does not create new alleles, so number 1 is wrong. It does create new combinations of alleles, so number 2 is correct. You do not need to know this one. This is important for evolution, that's why I bring it up. Remember how I said that there's 2 different types of reproduction. There's asexual and then there's sexual. Well, there's advantages and disadvantages to both. So let's evaluate sexual reproduction first. Advantage: sexual reproduction leads to offspring that are genetically different from each other and from either parent in 3 different ways. The first was combining alleles from 2 parents at fertilization. So that was one way of introducing genetic diversity. The second was crossing over events between those homologous chromosomes to produce the gametes. And then another one was shuffling and reassortment of homologs during meiosis. You're not going to understand number 3 unless you go through all the steps of meiosis, which we did not. Okay, but then bacteria reproduce asexually. So the variability among the offspring produced by sexual reproduction enables populations of organisms to better cope with change in their environment. After all, if the environment is gradually changing from one generation to the next, individuals producing many offspring increase the likelihood that one of their offspring will carry a set of genes particularly suited to the new environment. It's like buying lottery tickets. The more different tickets that you buy, the more likely one of them will be a winner. You don't buy all the same ticket, right? That wouldn't make any sense. Over time, populations of sexually reproducing organisms can quickly adapt to changing environments. Okay, so those are advantages of sexual reproduction. There are sex differences in the chromosomes. Alright, so we have 23 chromosome pairs, remember? 23 from our mom, 23 from our dad. Only one of the 23 chromosome pairs determines sex in humans. Okay, so you can be XY or XX. That's going to determine if you're male or female. In humans, the sex of a baby is determined by its father. Alright? So the eggs always have an X chromosome, but the sperm is going to either have an X or a Y. Alright? Okay, so let's look at this a little bit closer. The human sex chromosomes are called the X and Y chromosomes. How do the X and Y differ from the other chromosomes? As we described earlier, all of the genetic information is stored on the chromosomes in all cells of an organism's body, but most of the information is not sex-specific. That is, if you're building an eye or a neuron or a skin cell or a digestive protein, it doesn't matter whether it is for a male or for a female. The instructions are the same. Some genetic information, however, instructs the body to develop into one gender or the other. That information, which does differ dramatically, depends on whether the genetic information for building a female or a male is found on the sex chromosomes. An individual has 2 copies of all the non-sex chromosomes. 1 copy is inherited from mother, 1 from father. Individuals also have 2 copies of the sex chromosomes, but they vary a bit in how they do this. Males have 1 copy of the X and 1 copy of the Y. Okay, males are XY. Females don't have a Y chromosome, but have 2 copies of the X. Okay, so this is just showing you how the father's sperm cells determines the sex of the baby. Because the sperm can have either an X or a Y. The probability that an egg will contain an X chromosome is what percent, but in a sperm is what percent? So the answer is, of course, number 3. An egg will 100% of the time contain an X chromosome to pass on, but the sperm cell will contain an X only half the time. Okay, these are just summaries. You've probably heard of Down syndrome. Okay, so Down syndrome is a change in the chromosome number, specifically the chromosome number 21. It's when you have 3 pairs of it. The chances of having an incorrect number of chromosomes increases as a woman gets older. That's why there's a correlation between a mother's age and the risk of Down syndrome, because again, that is a duplication of chromosome 21. So you have 3 of them instead of 2. The karyotype is the entire chromosome set. That's all that word means. And it's defined here. We look at karyotypes to find out whether the offspring may have a disorder associated with incorrect number of chromosomes. A karyotype can be made for adults or children, but it is most commonly done for a fetus. A karyotype is a useful diagnosis tool because it can be prepared very early in the fetus's development to assess whether it has an abnormality in the number of chromosomes or in the chromosome structure. And because the test shows all of the chromosomes, even the sex chromosomes, it can also reveal the gender of the fetus. Okay, so that's how we determine that when we look at a karyotype. So what is the gender of this fetus? This is a karyotype right here. It is male because see at the bottom right there's an X and a Y. So here you're seeing the 23 pairs of all the chromosomes. And they are indeed in pairs. But if we had a karyotype for somebody with Down syndrome, if you go down to chromosome 21, we'd have 3 of them. Amniocentesis is just a procedure. It's done approximately 3 to 4 months into pregnancy. This is when a needle is pushed through the abdomen into the amniotic sac, into the amniotic fluid. That's just the fluid that surrounds and protects the fetus. They can take out that fluid and get those cells from the fetus to figure out the karyotype. CVS, or chorionic villus sampling, is a procedure that rather than sampling cells from the amniotic fluid, a small bit of tissue is removed from the placenta itself. The placenta is the temporary organ that allows the transfer of gases, nutrients, and waste products between a mother and fetus. So that's what the placenta does. So this is usually done between the 10th and 12th week of pregnancy. So here's somebody with Down syndrome. It has some distinctive phenotypes, as you can see in the photo of this gentleman. And the genotype is shown in the karyotype here, where you have 3 of that chromosome 21. It's striking that about 1 out of every 1,000 children born have Down syndrome. It includes learning disabilities, flat facial profile, heart defects, and increased susceptibility to respiratory difficulties. Down syndrome is one of the few times that you can have a change in the number of a chromosome pair and survive. So most of the time, if you will never hear about somebody having 3 copies of chromosome 1 or 2 or 1 copy of chromosome 3. Those things don't happen because that baby will never be born. It will be so dramatically messed up as a fetus that it will never keep dividing and become an entire human being. But 21 is one of the rare ones that can survive. What causes this messed up chromosome number, as an example, is non-disjunction. So that's when the chromosomes don't separate correctly when they're splitting into 2 cells or 4 cells like they're supposed to. So it creates a gamete with 0 or 2 copies of a chromosome rather than a single copy. So what is the probability that a baby born to a 45-year-old woman will have Down syndrome? Okay, so if we look at the x-axis, we go to 45, we go up. It looks like the chances are 20 per 1,000 live births. So number 5 would be the answer. Okay, karyotype displays a complete set of chromosomes, so you can get the number for each pair, make sure it's normal. Useful diagnosis tool. Alright, so you can have a change in the sex chromosomes and survive pretty well. When you have a change in those somatic chromosomes, the chromosomes that affect all the cells in your body, all the characteristics of your body cells, it's more disastrous. Okay, so Down syndrome is a surviving disease it's a disease where people can survive with a different number of chromosomes, but most of the time, like I said, it's not going to be the case. Those individuals will not survive to infancy. But if you have a change in the chromosome pair 23, the sex chromosomes, it's not fatal. These are the different syndromes. They have different characteristics. You can have an X alone in your sex chromosomes, or you can have an extra X, or you can have an extra Y, or you could even get 3 X's. All of these happen in a non-disjunction event, similar to, we talked about non-disjunctions for chromosome 21. Okay, so although it is usually fatal to have 1 too many or 1 too few of the non-sex chromosomes, Down syndrome being an exception, individuals born with only a single sex chromosome, that is an X, or with an additional X or Y chromosome, usually survive. They do have some physical and or mental problems.